Thank you again for visiting. Thank you for everyone who's on the call and the webinar today. So I'll pass it off to Dr. Rowe, who's going to share her work with us today.
Wonderful. Thank you very much, Narosha, and for Algoma University for the opportunity to come and present some of the work that I've been doing at the Great Lakes Forestry Centre. I'm just going to share my screen. I apparently have my screen sharing disabled. Could I please have that enabled?
Absolutely. Our apologies on that. Just one moment, please.
I could do it with hand gestures, but it might be less effective.
Narosha, if you'd be able to promote her as co-host, it's not letting me. There you go.
Excellent. Thank you very much. All right. So today I'm going to use this opportunity to talk about the Asian longhorn beetle and tracking and tracing their invasion in North America. Now, before I begin, I do want to acknowledge the work that has been done by Ming Ming and Ilka Porth at Laval University. Ming Ming is a PhD student who is working with Ilka and I will be presenting her work here today. So as many of us are aware, invasive species have had an immense impact on Canada's, North America's forests, both in the urban canopy and in the forested landscapes. And this picture here really highlights that impact, and our forests have experienced devastating invasions from, say, the emerald ash borer, which have targeted ash trees, the chestnut blight, which destroyed our chestnut forests, as well as Dutch elm disease, and that rid many eastern cities of their urban elms. So invasive species are a threat that are continuing and are not releasing. So we need to develop approaches and tools to be able to address this threat into our forests. But invasion isn't an instantaneous event. Invasion and movement of organisms into novel environments go through a series of stages, from transport, introduction to establishment and spread. And all along this invasion pathway are barriers and events which will leave a signature in the population. However, traditional methods of tracking and tracing invasions, looking at presence and absence of organisms, generally doesn't have the resolution to be able to get at at many of these stages and the underlying processes that are permitting invasion to occur. However, with the advent of new genomic techniques and tools, we have the ability to use the great wealth of information contained within an organism's genome to start to answer some of the questions around this process of invasion, such as where is something coming from? What is its history of spread and the risk that it'll establish, or its probability of survival in a novel area? And it's the ability to use genomics as a biosurveillance tool that underlies a large Genome Canada project that I am part of called BioSafe. And the goal of BioSafe is to, we partnered with the Canadian Food Inspection Agency, which is the main regulatory body who manages and controls invasive species in Canada, to identify a series of forest invasives that pose high risks to Canada's forests and develop surveillance tools to be able to respond rapidly and effectively to invasions by these pests. And one of the species that was identified as a high risk is the Asian longhorn beetle. And from now, I'm going to present some of the work that we've been doing with the Asian longhorn beetle as a part of this BioSafe initiative. So the Asian longhorn beetle is a large cerambycid beetle or longhorn beetle that is native to China and the Korean Peninsula. And in its native range, it feeds on a wide range of hardwood tree species, such as poplar, willow, and maple. In North America, it was initially discovered in New York City in 1996, and since then has spread throughout North America with a number of invasions, as well as in Europe. And in this invasive range, particularly in North America, the Asian longhorn beetle poses a significant threat to maple trees. And in Canada, we care quite a bit about our maple trees, both as an emblem on our flag, as well as the producer of maple syrup. But to really understand the process or the story of the Asian longhorn beetle invasion, we need to go back several decades to its native range in Asia. So in the 70s, climate change had already started to impact parts of northern China, and the Gobi Desert was expanding and starting to encroach on arable land along its along the Gobi Desert southern border. So China began what was called the Three North Project, or the Great Green Wall. And the goal of this was to plant a barrier of trees between the Gobi Desert and that arable land in the north, and basically reforest or reclaim land that was lost to this desertification. So it was a massive undertaking. In the 70s and the 80s, it cost $8 billion and resulted in 66 billion trees being planted along that encroaching edge of the Gobi Desert. The trees that were selected were fast-growing, drought-resistant trees and cultivars of poplar and willow. Now, these trees are a favored host of the Asian longhorn beetle in China. And since these trees were planted in really stressful conditions, the Asian longhorn beetle is able to get in and heavily infest many of these trees. The trees, when they matured, were actually harvested, but because the wood quality was quite low, it was turned into wood packaging material. Things like pallets or dunnage that are used to carry goods or package goods when they're shipped and traded globally. So the Asian longhorn beetle is a bit unique because it actually feeds on the inner core of its host tree as opposed to along the outside edge near to the bark. And so when wood is processed and the bark is stripped away, it actually still leaves that larvae inside of the tree. And they are quite long-lived, and so they were able to stay inside the wood and then would emerge when the pallet or with packaging material ended up in its place of, it ended up in its destination. And when the beetles emerged, they were in a novel environment and would seek out host plants in that novel environment. And so one of the key questions that we really want to address is where in its native range are these insects coming from? Because if we can understand where it's coming from, we can help to inform border surveillance as well as trade regulations for particular regions of that invasive from its native range. And this can start to allow us to dissect or tease apart the process of invasion, particularly for some place like Toronto, because we in 2003 ended up with an Asian longhorn beetle infestation in Toronto near the city of Vaughan. And we couldn't tell where this, what the source of this invasion was. Now, to be able to use genomics to understand and to trace the sources of invasive species, it really is predicated on the fact that there's an assumption that there is population structure throughout the native range. So as an example, if we had an Asian longhorn beetle populations in its native range that were grouped into distinct variants. So here in this map as an example, we had a blue variant in the north, a green variant in the west, and an orange variant in the south, and then a red variant in South Korea. Then we could use that genetic variation to trace new intercepted beetles back to its native range. However, if the native range looks more like this, where everything looks similar, there isn't enough information there to be able to resolve where things are coming from. And so the first step in this process is to delimit or define the population structure in the native range. Some previous work has been done on this question, but the projects that had initially been performed used markers that maybe were not variable enough or widespread enough throughout the genome to give us amount of information to be able to resolve those population level differences. And so a publication in 2009 and another in 2018 focused on using microsatellites and mitochondrial DNA to be able to resolve the populations. And while we see some structure, there's a great deal of complexity within the data set. And it may be that these markers are not appropriate for this question, or that there has been contemporary movement of Asian longhorn beetle in its native range that has caused mixing among those populations, blurring those population boundaries. But to be able to address whether this is what's going on, we needed to look at that question using some more finer resolved markers. And so this is the work that Ming is doing at the University of Laval, and we are using new genomic techniques to be able to resolve genomic or genome-wide markers spread throughout the genome of the organism to quantify the genetic variation in the native range and then place the populations in North America into that context. And so to do that, we had a number of collaborators throughout Europe as well as in China help us to collect individuals throughout the native range in China. And then we used a whole genome sequencing or genome-wide marker selection to be able to select SNP markers and then quantify population structure and genetic diversity. And so to start, I will present the genetic variation, but really I'm going to present a simplified version of that in the form of principal coordinate analysis. And really all you need to understand is that this is taking a lot of complex genetic variation and reducing it down to 2 axes. And things on this little dots represent individual beetles, and dots that are close together are more similar to each other, and dots that are far apart are different. And the further apart you are, the more different you are. And so to start, one of the big questions is, can we resolve differences between China and South Korea? And what we find is that, in fact, yes, in the blue circle here, you can actually see, I'm just going to pull up a mouse. In the blue circle here, we can see that there is 3 individuals. This represents the South Korean population, but we also see here these blue arrows represent individuals that were collected in Korea that fall closer to individuals that are from China, the Chinese mainland. And these are what we take as evidence of contemporary movement of Chinese beetles moving into the Korean population. And we're seeing that level of resolution in our data set. If we look a bit more closely at the individuals that we find in China, we can see that there is fairly clear population structuring among the populations. And the big takeaway here is that we do see a northern population and groups of populations and a southern population. And those are represented here by this cluster on the right, and then this long vertical cluster here on the left. And if you look even closer, you can see that those bigger north and south populations are also show geographic structuring. In particular, we see a latitudinal gradient along the east coast that aligns with climate. And this is very interesting here. And so if we cluster this variation using a discriminant function analysis, we can see that the populations group into 6 main clusters. So we have here in blue individuals in the northwest, we have in green the central populations, 2 populations up in the north, the purple and the peach, and then 2 populations along the eastern coast. So now that we have resolved these regional population structures, we can begin to ask, where are our North American invasive populations coming from? And so what is that history of spread in North America, and can we resolve the source of our invasions? And this could be really important when we look at places like Toronto or other invasive areas, because with Asian longhorn beetle, eradication is the main form of control. And one of the questions you want to be able to answer is whether an eradication was successful. So in Toronto, our initial detection occurred in 2003, and that population over the next 7 years was eradicated. But after 6 months after declaring that initial Vaughan population eradicated, an additional detection in 2013 occurred outside the city of Mississauga. And so one of the really important questions is whether the Mississauga population was in fact a satellite of the initial 2003 Vaughan population, or was this a new invasion? And genomic tools can help us address that question, because it really helps to tell the difference between, from a management perspective, as to whether it was the survey process that had missed that initial satellite, or was it a new invasion that was missed during border detection. And in Canada, we've only had the 2 invasive populations in Toronto, Mississauga. In the States, there have been a large number more. We've had infestations in New York, New Jersey, Boston, Worcester, Chicago, and Ohio. And this is still an ongoing problem, because in October of 2020, a new find was found in South Carolina outside of a golfing community, and there are thousands of infested trees in the forests outside of this community. And so it continues, Asian longhorn beetle does continue to pose a threat to our forests. And so using the same genomic approaches that we had used for our native range, we quantified the genetic variation of our North American populations. And we can see that there is significant structuring among these North American populations. So we have a large group of individuals that come from New York, New Jersey, Massachusetts, and Illinois. So these all are genetically similar. Ohio, and then one population out of New York in Farmingdale show a fairly distinct signature. And then we have our Toronto infestation. And what we can see here is that our initial 2003 find is very similar to the 2013 satellite in Mississauga. And so we take away from this is that we're quite confident that the 2013 satellite was indeed a missed population during the initial surveys of and the eradication of that 2003 find outside of Vaughan. One other piece of evidence to suggest that this was an isolated satellite is that it was a lot of siblings. And so with these genomic tools, we can actually quantify kinship. And many of the beetles that were sampled in that 2013 location were related and quite close related. And so that gives us another piece of evidence to suggest that this was indeed a satellite. So where do these populations now, how do they relate to the genetic variation that we see in the native range? And so just to remind you, we had the 6 discriminant groups on the left-hand side here, and then we have our on the right, I'm showing you in the colors, the invasive populations out of North America. And so first off, we can see that here are those native populations overlaid onto the principal coordinate analysis. And we can see that most of our native populations in that blue group here, so that includes New York, New Jersey, and Illinois, are most closely associated with either the northwestern group in light blue or the dark green group in the central in the central area. However, there is some interesting patterns that seem to fall out. So Massachusetts appears to have individuals of multiple sources, suggesting that there may have been a mixed invasion in Massachusetts, and some additional work needs to be done to look at the source of this individual here from Massachusetts. But Ohio and Toronto appear to be still falling outside of the amount of variation that we're seeing in the native range. And so the question is, why are we seeing such differences in these two populations? Well, if you start, you have to remember that when we present this information in a principal coordinate analysis, there are multiple dimensions to this data. And those first two axes represent the maximum amount of variation, but we can actually start to look at additional axes and drill down through that variation to see where Toronto and Ohio start to associate if we remove that sort of unique signal. And you can see that Toronto and Ohio both group with these populations along the eastern seaboard. And so what we take from that is that the North American invasion was indeed from multiple sources throughout from its native range. And it wasn't a single source. And so it is important from a trade perspective to understand the sources of these invasions. But why was Toronto and Ohio so different in those first two principal coordinates? And it goes back to that process of invasion, because invasion can have things like founder effects, where a single individual or a small number of individuals invade, and that changes the genetic composition of the invasive population. And this may be just due to neutral variation that doesn't have an adaptive advantage or adaptive basis. The other issue is that there may be a process of adaptation, and that these are populations that were selected over a period of time. Now, it is important, however, to recognize that Asian longhorn beetle has only at the most one generation per year, and so it's a very short period of time for adaptation to have occurred. But it is certainly something worth exploring, and we're continuing to delve into this question. And so from our preliminary results on the history and spread of the North American Asian longhorn beetle invasion, is we are confident that there were at least 2 or 3 distinct sources in North America. And Toronto and Ohio came from the eastern coast of the native range. And then there was populations from northwestern China and potentially from the central regions. And the Toronto and Ohio infestations represented fairly distinct populations compared with the other invasions in North America, and may be evidence of either founder effects or of selection. And then finally, we were able to show that the 2003 and 2013 infestations in Canada had the same genetic signature and were likely from the same initial invasion event. And just to put this into a global context, there have been a number of invasions in North America, but Europe has seen significantly more invasions throughout many countries in Europe. And they have been investing many millions of dollars to eradicate these populations. And the eradications have been successful. So we were actually able to declare Canada Asian longhorn beetle free this spring, actually in June of this year, or of June of 2020, as well as a number of other infestations in Europe. But we should also acknowledge that there is continued detections of new invasive populations, such as what happened in South Carolina in 2020, and even in Canada, we had an interception of a live Asian longhorn beetle coming out of wood packaging material in Edmonton, and so it is a continued issue. But looking at this timeline and this map of all of these different invasions, one of the things that I want to drive home is that nearly 90% of these detections of these new invasions were done by private citizens. And private citizens have been critical in the response to Asian longhorn beetle. And if you look at this invasion curve, the earlier you can get or identify an invasion, the more likely you're going to be successful in eradicating and preventing the spread of a new invasive species. We have the advantage with Asian longhorn beetle of a very large, charismatic, distinctive insect. Its damage is distinctive as well as the adult form. And they're both in Canada as well as in the US, there was a major science communication push to be able to present lots of information for the general public to help them identify Asian longhorn beetle if they see something. And it comes back to having a public that is engaged with the local environment and acknowledging that when they see something unusual. And as you can see, it is a very distinctive insect, and these damage holes are quite large and are easy to spot. And so having clear pathways of communication for publics to be able to reach experts and to notify the authorities of potential invasive species is critical. And so to conclude, this part of my talk is that we were able to use genomic tools to demonstrate that native Asian longhorn beetle populations are structured, and that these genomic tools can identify source regions in the native range, and that these North American populations are distinct, and we were able to associate them with known regions in the invasive range. We were able to demonstrate that the Toronto population in 2003 and 2013 had the same genetic signature, and that private citizens really are critical in the detection of new infestations. All of this work is due to a collaborative effort both of many students and researchers, as well as collaborators throughout Europe and in China. And it's really through all of their hard work that I was able to present this work here today. Now, before I move on to questions, I do want to take this opportunity to highlight some of the broader research projects or the broader research expertise that exists in my organization. So as was mentioned before, I work at the Great Lakes Forestry Centre, and it is only 2 or 3 blocks down the road from Algoma University. So this is a large federal research building. It has been in Sault Ste. Marie for 75 years. Actually, 2020 was our 75th year of forestry research. And it focuses on sort of 6 major research themes. And I'm just going to highlight those here quickly. So we have a lot of research on forest ecosystems and forest ecosystem integrity. There are 8 research scientists associated with this research branch within our organization, and they use biodiversity to be able to assess ecosystem integrity. And we do this both within the terrestrial ecosystem as well as in the forest soils and within the riparian and water throughout the forest zone. Now, one of the critical some of the critical infrastructure that is maintained by the Great Lakes Forestry Centre is the Turkey Lakes Watershed, which is a long-term research site that is north of the city, and it was initially established in the 1980s to study impacts of acid rain on the forest ecosystem, but has since expanded to look at a wide range of disturbances and pollutants, and represents a great deal of long-term data sets as well as site history, and is a very close but valuable research site. Fire is another big part of our research program. We actually have 9 research scientists who are focused on fire and fire research. And the science at the Great Lakes Forestry Centre was responsible for the development of the fire danger, the Canadian Forest Fire Danger Rating System. And so it's the little dial you see if you go to a park to tell you whether you're allowed to burn or not. That was work that was all completed at the Great Lakes Forestry Centre. There's also been a lot of science to understand the risks and the hazards forest fires pose to the urban landscape, and especially at that urban wildland interface. And most exciting, we actually have a research scientist who is leading the development and launch of a wildfire satellite, which is a dedicated near-Earth satellite, which is going to provide forest fire remote sensing and research. And this is in partnership with the Canadian Space Agency. And they're hoping to be able to launch this wildfire sat in 2024 or 2025. So this is really, really exciting stuff. So the unit that I'm a part of is the Integrated Forest Pest Management Unit. And this is probably the largest research unit within the institution, which is probably why GLFC earned the nickname the Bug Lab among the locals. We have 12 research scientists associated with our unit, and its research here is split into sort of 2 main focuses, both insects or molecular biology and pathology, and then forest pest ecology and pest management. And I really straddle those 2 fields with my interest in molecular ecology and functional ecology. And so we're trying to understand the molecular mechanisms underlying insect function and diseases, and as well the population dynamics, as well as management and control and surveillance for both native and invasive pests. One of the critical pieces that of research infrastructure that we have is the insect production and quarantine laboratories. And this is a facility that maintains and rears large numbers of insect colonies, as well as a quarantine lab for working on invasive species. I lead this unit, and we provide support for research both within our organization as well as external organizations throughout the world. It's also the main, the key player in Canada's EA Emerald Ash Borer Biological Control Program, and we rear Canada's only parasitoids, which are used to try and control the emerald ash borer throughout Eastern Canada. We have what's called the Canadian Wood Fiber Centre, and this is a group of researchers who are focused on understanding wood and wood properties, as well as adaptive silviculture. And this is basically trying to understand how to develop and grow forests in a sustainable way. A lot of their work is done at the Petawawa Research Forest, which is actually Canada's oldest continuously operating research forest. And this is out in east towards Ottawa. And it's considered a living laboratory and has some of the oldest permanent site plots, forest plots in Canada. And is an incredible place to do research, given the research history and the site history that we have records for. We have a spatial analysis and economics unit with 2 research scientists, and their work has really focused on understanding spatial variation of a number of different, from a number of different angles, whether it's from forest productivity or species distributions, both of native organisms as well as invasives. They are also responsible for the development of Canada's plant hardiness zones, which you can see here on the left. And their work on climate and climate modeling has been integral for much of the climate change research done in Canada, both within the federal government and within universities. And finally, underlying all of what we do is the interest in engaging with our Indigenous partners. And there's been a focus on getting research scientists engaged and increasing our awareness of Indigenous ideas and Indigenous knowledge, and engaging with that Indigenous traditional knowledge and looking to integrate that within the forest science that we conduct. And supporting our Indigenous neighbors and the Indigenous communities to try and build capacity within their communities, both from a scientific or knowledge perspective, as well as from an economic perspective. And so with that, I will welcome questions, whether it's on my work on the Asian longhorn beetle, or if you have any questions about the type of work that is done at our research institution. And
Thank
so
you.
with that, I will finish.
Thank you so much, Dr. Rowe. It was a really comprehensive talk, and I think I particularly enjoyed the ways students can get involved with all the various networks that the Environmental Center has to offer.
Yeah.
Thank you so much for that. So I will open the floor to any questions from our audience members about her research, as she said, or about potential opportunities for research experience or employment. It is lunchtime, so I know.
Yeah.
Oh, we do have a question here from Dr. Bloomfield. The bug shown on the slide with the insect production and quarantine laboratory sign appears to be what I call a pine beetle. Are these beetles and the longhorn related? What is the closest relation to longhorn? Good question. Cool.
Yeah. So it's a silhouette of of a longhorned beetle, and so the Asian longhorn beetle is in the family Cerambycidae, and what people here call pine bugs. The scientific name would be Monocamus scutellabus, which is the most common one around here, or the pine sawyer beetle. They would be in the same family. And so they are related. The pine sawyer beetle or the pine bug are native to Canada and feed in dead or dying softwood trees, so conifers, whereas the Asian longhorn beetle, is an invasive species.
Awesome.
So while we're waiting for questions to come in, I do wanted to mention that there is a really great opportunity and a way to get involved with federal research science, not just within the Great Lakes Forestry Centre, but among all of the different federal research labs that exist across the country. And that's through the FSWEP program, which is the Federal Student Worker Experience Program. And there is, if you look up FSWEP, you can actually submit your application through that program to gain summer employment within the federal research environment. And this is the main way that our organization hires student workers. And right now is when you need to be getting your applications in, because we are in the process of hiring students as we speak.
That's awesome. Really good to know. I think I'll highlight this on the Research Week website, and I'll send this with our Career Center, who will also highlight this as well for
That would be
great
great. Yeah.
job experience. We do have another question here from Jonathan. Is there any genetic data on the types of trees that may be more or less affected by the Asian longhorn beetle?
That's a really interesting question. We have some evidence that there seems to be a preference in North America for maples, based on where we find the beetles when they are in an invasive population. But very little has been done to look at whether individual trees show a resistance to the beetle, or whether one sort of strain or genotype of the tree shows a resistance to the beetle over another. And part of that comes from the fact that we have the management approach for Asian longhorn beetle is eradication. And in Canada, that is cutting down and completely destroying any trees that could have the beetle in it. And so it's been, less work has been done on the Asian longhorn beetle for that question. However, for the emerald ash borer, which is also an invasive beetle, more work has been done to try and identify ash trees that show resistance to that invasive pest, with the hopes of selecting and reforesting with a more invasive ash community, because of the importance ash trees have within our forest ecosystems.
Awesome. Hope that answers the question. We have another question from Dr. Antunes. Is there any inventory of pallet factories in China and Korea? It seems like this would help identifying with insect sampling collection and source tracking. Yeah, for sure.
Getting that type of information has been very challenging, and the diversity of like I presented pictures of pallets, but it's really any wood packaging material, and there are thousands of forms that can take. It is quite challenging to be able to trace that to individual companies. It is something that is being explored, because if you can at least identify the source, like if you can find the initial packaging material, there usually is some identifying features that could help guide you to a source. But often the infestation is found years after the initial escape from the trade goods that they were associated with. And so we don't usually have that information at that point in time. And like any supply chain, the wood may be lumbered in one place and manufactured in another place, and so the supply chain isn't as simplistic as individual factories.
I had a question to kind of piggyback off Dr. Antunes' question. Are there any policies or restrictions that you can think of that are made at the manufacturing level that could prevent the pests from surviving on these materials, that would prevent them from invading?
Yeah, and so actually in the 90s, when wood boring insects were identified as high risk invasive, and we started to identify the wood packaging material pathway as really high risk, there was an international initiative to develop what's called IPSM 15, which are heat treatments that are applied to wood packaging material that will kill anything that lives within or on the wood prior to shipping. And IPSM 15 certainly has improved the control of that invasive pathway. However, it's not 100%. So sometimes the treatment isn't 100% effective. There are occasions where companies will fake the treatment, because any wood packaging material that has been IPSM 15 certified has to receive a stamp, and there are there have been fraudulent stamps applied to wood that has living organisms in it. And so even with these added barriers, there is still a risk to that pathway, and we need to stay vigilant with our surveillance at borders as well as with private citizens.
Awesome. So is there something that consumers like us that we can do when we receive these packaging? Is there, like, in terms of getting rid of, destroying, recycling?
So it's not usually private citizens that are receiving pallets. Like these are pallets that are arriving at Walmart, and all of your goods are stacked on top of the pallets. And pallets are everywhere. Like you look at it behind every grocery store, every store. This is just how goods are moved. And it's a sustainable way of moving goods, because it's wood, and wood is better than many other forms of materials. The best thing we can do is to be out in the environment and be aware of what is native and what might be unusual. And when you see something unusual, take a picture and tell somebody, because every one of us has a camera in our pocket now. And so there are lots of places where you can reach out to. And one of them actually is here in Sault Ste. Marie, and that's the Invasive Species Centre. And the Invasive Species Centre, its entire focus is connecting the general public with the knowledge of invasive species and how to identify them. And if there is a sighting of something unusual, they know who to contact, and they act as a knowledge broker to be able to connect the general public with people like myself or other researchers who are experts in this field. And so these are the types of people that the general public can reach out to help them if they see something that looks a little different or looks unusual. And it's those eyes on the ground that are so important when we talk about invasive species, because reacting early is the best solution, and being able to respond quickly is really dependent on that initial early detection.
Awesome. We've got a couple questions here coming in. Apart from the threat to the maple trees, does the invasion of the Asian longhorn beetle pose a threat to any other beetle species population that are native to Canada?
We haven't seen any evidence that they are posing a risk to other beetles specifically or other organisms specifically, other than the fact that the threat posed to a tree species will inevitably impact a whole community of organisms, and because that species of tree is an integrated part of a larger ecosystem. And as we've seen with the loss of our ash trees or our chestnut trees or our elm trees, the loss of a single tree species can have widespread devastating impacts on all sorts of communities, and many of which we are not able to predict until we have lost that member of the community.
I think we have one more question here from Anal Patel. Apart from being, that's the same question. So I hope that answers that. Do you have any other questions from our audience? And as always, if you do have any more that you think of after today's session, feel free to email Dr. Rowe directly or the research office, or use the contact form, and we'll make sure that Dr. Rowe gets those questions.
Absolutely.
Yeah, I'm not seeing none, but again, I'll take this opportunity to thank you again, Dr. Rowe, for coming in, sharing your work, sharing what the centers do, and more importantly, giving students this opportunity for experience and employment. I think that's super exciting and important. Great timing too, especially if they're hiring right now.
Yes, absolutely. So thank you, and yeah, thank you for sharing the link, and absolutely be sure to apply. And there's lots of opportunities federally, locally, but then also across Canada. So lots of great opportunities out there.
Great. So I think this is a great keynote session. Again, I'd like to take this opportunity, everyone, to thank Dr. Rowe. And tomorrow is our last faculty lunch block talk, so I hope to see everybody there.
Great. Thank you very much, Narisha.
Thank you. Thanks, everyone.